Hi, everyone. Welcome back. Hope you all are well today and your families. Just an announcement before we get started today to please read your emails and announcements. I have sent out several, and they contain a lot of information about how we are going to run the rest of this course, including deadlines for your assignments, etc. So please just check your email regularly. And also, I did post announcements for a final wrap up. I am also going to be on live chat through Canvas on Tuesdays from 2 to 3. So if you want to chat with me directly, it's just like any messenger type setup. You just go to Canvas on the left hand menu, click chat, and you can talk to me directly, or of course, you can always email. So, with that said, we are today going to chapter 17, which begins on page 335 in the text and page 103 on the course outline. And here we're going to continue our discussion of DNA. And we have already talked about the fact that DNA forms chromosomes, which carry genes that code for proteins. So in this chapter, we're going to see exactly how those genes are converted to proteins in a process called protein synthesis. So hopefully you remember synthesis means building, and so we are actually building proteins. So genes, of course, hopefully again you remember, are pieces of DNA that code for these proteins. And when we talked about genetic traits, really those traits are specifically proteins. And I've said that many times. But this time, instead of just focusing on DNA, we're also going to add RNA. And RNA is also a nucleic acid. It's also made up of nucleotides, just like DNA. But the big difference is DNA has a D and RNA has an R. So the R in RNA is for the sugar ribose. In DNA, it's deoxyribose. But RNA is actually a helper. And I brought in a little humor again from Amoeba Sisters, and they're talking about RNA as a helper in a kitchen, and it says if you can't help make proteins, get out of the kitchen. So that's a pretty good analogy, and that's exactly what RNA is. It's a helper to help make proteins. But in another analogy that I really, really like is using this idea of building a house or constructing something. And so this is an old transparency I've had forever, and I can't find anything else like it. So I took a picture of it because it's a great analogy, and I also have this on Canvas as a handout. But anyway, it shows you here, if you start with DNA, and if you remember, DNA is usually locked up in the nucleus. The only time it leaves the nucleus is when the cell is ready to divide. But most of the time, then, it's stored at what we could use an analogy of the headquarters. So let's say you want to build a house, and you have a blueprint for that house. You don't want to take the original out to the job site. So the original is DNA, which provides the instructions, but instead you have this mobile messenger RNA that's going to take it out to the job site. The job site in this case is the cytoplasm, and that's where the workers, which if you remember, ribosomes are involved in protein synthesis. So these workers are at the construction site, and they are going to work along with uh, amino acids. Remember, amino acids are the building blocks of a protein, so those are actually the workers. The ribosomes are going to link those all together to make proteins. So the final product is the protein or the house. What kind of proteins do we need to synthesize? Many, many different kinds of proteins exist, and we've talked about this. These are a very diverse group. They include structural proteins like the keratin of your hair, your nails, organs, organ systems. All the structural stuff in our bodies are made of proteins. And then we have functional proteins, those that have a job to do, like hemoglobin. And as we've mentioned many times, hemoglobin's job is to carry oxygen in the red blood cell. And then remember, all of our enzymes are protein based. So all of our metabolic reactions require enzymes in order for those to happen. So many, many different diverse types of proteins that we need to synthesize. And we need to do this very rapidly and very often in every cell. So this idea of protein synthesis is hugely important. So if we look at the overview of this process, the information content of DNA is in the form of specific sequences called nucleotides, and the DNA inherited by an organism leads to specific traits by dictating the synthesis of proteins. So proteins are the links then between genotype and phenotype. So the genotype is coded in the DNA, whereas it's expressed in our bodies as phenotypes. And so when we use the term gene expression, that is a process by which DNA directs protein synthesis, and it includes two stages, transcription and translation. So I want to play this clip that will talk about these processes, and then we'll go into each one specifically.
Listen up. The soap you shower with, it's you probably haven't even questioned what bar of soap you lather up with.
Nucleus. The nucleus contains. Here is a cell, the basic unit of all living tissue. In most human cells, there is a structure called the nucleus. The nucleus contains the genome. In humans, the genome is split between 23 pairs of chromosomes. Each chromosome contains a long strand of DNA, tightly packaged around proteins called histones. Within the DNA are sections called genes. These genes contain the instructions for making proteins. When a gene is switched on, an enzyme called RNA polymerase attaches to the start of the gene. It moves along the DNA, making a strand of messenger RNA out of free bases in the nucleus. The DNA code determines the order in which the free bases are added to the messenger RNA. This process is called transcription. Before the messenger RNA can be used as a template for the production of proteins, it needs to be processed. This involves removing and adding sections of RNA. The messenger RNA then moves out of the nucleus into the cytoplasm. Protein factories in the cytoplasm, called ribosomes, bind to the messenger RNA. The ribosome reads the code in the messenger RNA to produce a chain made up of amino acids. There are 20 different types of amino acid. Transfer RNA molecules carry the amino acids to the ribosome. The messenger RNA is read 3 bases at a time. As each triplet is read, a transfer RNA delivers the corresponding amino acid. This is added to a growing chain of amino acids. Once the last amino acid has been added, the chain folds into a complex 3D shape to form the protein.
Okay, so looking at concept 17.1, where genes specify proteins via these two processes, transcription and translation, what you want to remember is transcription is converting DNA to RNA, and it happens in the nucleus, and then translation is converting RNA to protein. And often you're going to see messenger RNA emphasized as mRNA, the M for messenger. But there are actually 3 different kinds of RNA that are transcribed in the nucleus. In addition to messenger RNA, we're going to see there's transfer RNA, abbreviated tRNA, and ribosomal RNA, abbreviated rRNA. So we'll come to those later. But again, messenger RNA gets the most attention because it is actually carrying the message from DNA out into the cytoplasm where it will be translated into proteins. And remember, ribosomes are involved in all of this. They are out in the cytoplasm, and that's where translation occurs. So we have something called the central dogma of, of protein synthesis. And as you see here, what it is that we have these two major processes converting DNA to RNA, which is called what again? What? Transcription. And where does that happen? That happens in the the nucleus, and then converting RNA to protein, that's called translation, and that occurs in the cytoplasm. So two major processes that we're going to look at. Now, a lot of study on this began, as always, with bacterial cells because they're prokaryotic cells. They're much easier to study, and they don't have as much DNA involved as well. So when you look at transcription in a bacterial cell, we can see that here's your piece of DNA. Now, when you compare transcription to DNA replication, which we talked about in the last chapter, in DNA replication, all the DNA has to be copied and made for each new cell. In transcription, you're only copying a gene region of DNA. What is a gene? Again, a gene is a piece of DNA that codes for a protein. It could consist of 10 nucleotides. It could be hundreds of nucleotides. It just depends on what it's coding for. So here's your piece of DNA. Remember, it's double-stranded. It's going to be transcribed. Now, in a bacterial cell, remember, they don't have a nucleus. So this all happens sort of haphazardly within the cell. It's not compartmentalized like in our cells. But here's your gene region. It's converted to RNA. Remember, RNA is single-stranded, and that's essentially transcription converting DNA to RNA. Just one copy or one side of the DNA molecule is going to be copied because we only need one copy of RNA rather than, as we saw in DNA replication, where both sides were copied, so we had two, one for each new cell. In translation, we're going to take that code that messenger RNA has carried out to the ribosome, where it will work in order to bring up the correct amino acids. Remember, amino acids are the building blocks of proteins, bringing these amino acids up in order to make a polypeptide chain. What is a polypeptide? Remember, poly means many, peptide involved in peptide bonds, which join amino acids together. So a polypeptide chain, shown here as a beaded chain, these beads representing amino acids, is really just a protein. So again, this is much simpler in bacterial cells, and this is going to be important in our next chapter when we get to DNA technology, where we try to take human DNA, cut it out of our cells, and insert it into bacterial cells in order to get them to produce human proteins. So, for example, we might take the gene for insulin, the human gene, put it into a bacterial cell and get them to produce the protein insulin and use that as a pharmaceutical. So we can do that, but again, it's a little bit complicated because our DNA is a little different than bacterial cells. So we'll talk about that more later. But if you compare that to a eukaryotic cells, one of the big differences in human DNA and all eukaryotic DNA is that there are regions that are not coding for any particular proteins. We call this nonsense DNA. And these non-coding regions have to be removed before we can continue from transcription into translation. So we start out with the same thing, a piece of DNA, a gene region only, and it's converted to messenger RNA, but this is called pre-messenger RNA because it needs some final modifications. We have to process it, and we'll get into the details of that in just a minute. And then once we get our final processed RNA, we call it then messenger RNA. So the pre-messenger RNA has to be converted to messenger RNA. Now it's ready to go out into the cytoplasm where it will be translated into proteins. And again, this involves use of the ribosomes, messenger RNA, amino acids, and some other things we'll get to again in detail. So we have this genetic code, and if you look in your text, you will see the genetic code. I'll put this up in just a minute. And it's a series of these letters, A's, T's, G's, and C's, but we only have 20 amino acids, and we only have 4 letters to code for these amino acids. and only 20 amino acids to make up all these diverse proteins that we've been talking about. So how is that going to work? So think about this. Think about these 4 letters as having an alphabet with only 4 letters. And then if you take those letters and put them together in different sequences, you will get different words. And put those words together in different orders and you get different meanings. That's kind of how this works. So even though our alphabet only has 4 letters, if we mix them up and shift them around, we can code for these different amino acids, put those together in different sequences, and you get all the diverse proteins we're talking about. Anybody remember from watching that video that I just showed you how many nucleotides correspond to an amino acid? Anybody? Raise your hand and tell me. It's kind of weird talking to myself. Yeah, so there are actually 3 letters. We call it the triplet code, right? Triplet means 3. So here is our template strand of DNA. Remember, the word template means pattern for assembly of something else. In this case, we're taking one side of the DNA and copying only one side. That one side is called the template strand. So just like when DNA replicates, the DNA has to be unzipped to expose those free bases. But now in transcription, we're converting DNA to RNA. And the base pair rules are the same except one major difference. There are no T's in RNA. Notice instead of T's, you put a U. So here's our template strand. A brings up U, C brings up G, C brings up G, A brings up U, A brings up U, so forth. T brings up A, so there are A's. The only thing you have to remember is there's no T's in RNA. So the base pair rules are the same. In DNA, if you're making more DNA in DNA replication, it's A with T, G with C. But when we're making RNA from DNA, it's A with U, but still G with C. So we read these 3 letters at a time, and the 3 letters on messenger RNA that we read are called codons. And the reason they're called codons is because they code for an amino acid. So every triplet codes for a particular amino acid. So this one's tryptophan, UUU codes for phenylalanine, GGC codes for glycine, and so forth. So here is a look at that genetic code. So this is kind of our alphabet. And if we look at the bases, we have 3 bases. This would be the first base, this is the second base, and this is the third base. And if we want to see what amino acid they code for, we just look up the 3 letters. For example, let's look up AUG. If you look where all 3 intersect, here's A, here's U, and here's G, and that you see AUG codes for the amino acid methionine. This also happens to be what is known as the start codon. So we always start with AUG. Think about this. You can remember that because we start classes in August, right? AUG, that's our start codon. We have 1 start codon, and we have 3, as you see here, stop codons. These codons do not code for an amino acid. All they do is block the polypeptide chain from continuing to being produced. So these are just stop codons. Think of them as sort of like just scissors that stop everything, cut it off. They do not code for an amino acid. But notice we have 3 different codes, UAA, UGA, and UAG, all code for stopping the production of that protein. So some of these, if you'll notice, if you look at something like glycine, glycine can be coded for by GGU, GGC, GGA, or GGG. So that third letter can vary. In many cases, this is what is known as the wobble effect, and that the first 2 letters seem to have more effect, and that last letter in this case can vary, and you still get glycine being pulled up. So on to concept 17.2. Let's look at transcription. Again, transcription is converting what to what? So transcription is converting DNA to RNA, and it occurs in the nucleus. Now, as you look through your course outline, and I'm on page 104, you'll notice that both transcription and translation have 3 stages, and the stages are the same 3 stages: initiation, elongation, and termination. The difference is that different things are happening in each stage. So first, let's watch this animation of transcription, and then we'll get into the 3 stages.
A structural gene is made up of a sequence of bases in a DNA molecule consisting of a coding region with an upstream promoter and a terminator downstream of the coding region. The transcription of a gene into a messenger RNA sequence is initiated when RNA polymerase attaches to the promoter region and forms an open complex. RNA nucleotides are then polymerized from nucleotide triphosphates to make a messenger RNA. The messenger RNA sequence is elongated as the RNA polymerase moves down the DNA molecule until the RNA polymerase reaches the terminator region. Transcription is terminated when sequences in the terminator region are encountered. This causes RNA polymerase to dissociate from the DNA molecule, and the completed transcript is released.
Okay, so the first stage is initiation, and this begins on a part of the DNA called the promoter region. And as the name implies, it promotes transcription. And we begin with a sequence that is rich in the letters A and also T. And so it's commonly called the Tata box, as you see here. This is the promoter region. Keep in mind, the promoter region is a region of DNA, but what it attracts is this enzyme called RNA polymerase. So remember when we talked about DNA replication and converting DNA to more DNA, we needed DNA polymerase. But when we're converting DNA to RNA, we need RNA polymerase. Very important not to get those two mixed up. So what happens then with the promoter region is that several transcription factors bind to that region, and then transcription initiation complex will be formed. And essentially what that is all these proteins that attract this RNA polymerase, and that begins transcription. So here we have the transcription initiation complex formed, and that's what happens in initiation. Essentially everything loads up on the promoter, specifically the RNA polymerase. Not to be confused with DNA polymerase, right? Okay, now we're ready for step 2, and this is when we have elongation. So in elongation, what happens is the RNA polymerase shown here, remember that's an enzyme, it ends in ASE. Just like DNA polymerase brought up the DNA complementary bases, RNA polymerase brings up the RNA complementary bases. And it's only going to code on that template strand. So here's the non-template, we're not coding on this. This is the template. And so, as we've seen before, T attracts A brings up U. Remember, there's no T's in RNA. G with C, G with C, T with A, T with A with U, and so forth. And it continues on, and that is elongation. So again, taking it from the top, in initiation, we have RNA polymerase binds to the promoter region. It starts moving along in elongation and bringing up the complementary base pairs for producing an RNA transcript. And that is elongation. And at some point, it's going to reach a termination point, which is just the opposite of the promoter. The promoter attracts RNA polymerase. The termination point causes the RNA polymerase to be released, and now you have your completed RNA transcript. And it's ready to go. So there's your 3 stages: initiation, elongation, and termination. Now there's a problem, though, in eukaryotic cells, right? Because this RNA transcript still has that non-coding region, or those non-coding regions. And so we have, when we look at this RNA transcript, we see it contains both introns and exons. Exons are going to exit. They're involved in the next stage, which is translation. But introns are non-coding, and they have to be cut out. So this is our pre-messenger RNA transcript. Once the introns are cut out, the exons will be spliced together with using some enzymes, similar to what we saw in DNA replication, which we called the DNA, or some of those enzymes were like DNA scissors, some were like DNA paste. And then we have two more things added. On one side, we have something called a poly A tail. That stands for poly meaning many, right? The A is adenylate acid. So there is a group of adenylate acids added to this end. No one knows for sure why, but perhaps these modifications happen because it may protect it when it gets out into the cytoplasm from being broken down. So we're on page 106 of the course outline and looking at how these modifications take place. Now on the 5' end, there is a cap added called, which is a guanine nucleotide. And again, not sure exactly, but perhaps these final modifications are helping it to stay together when it gets out into the cytoplasm. RNA splicing then removes introns, right, the non-coding regions, and joins exons, the coding regions. So looking at the RNA transcript here, how do we get those introns out? And we use a variety of proteins and several small nuclear, what are known as ribonucleoproteins, called snRNPs, that recognize these splice sites. And notice that they attach to either end of the intron, and they cause this loop, and then enzymes will cut that loop out. So here's your cutout intron, and other enzymes will splice it back together. So here's how this works.
Most eukaryotic genes are composed of numerous short coding sequences called exons, embedded within stretches of non-coding sequences called introns. The initial messenger RNA molecule, or primary transcript copied from a gene by RNA polymerase, is a faithful copy of the entire gene, including introns as well as exons. Before the primary transcript is translated, the introns are removed by a process called RNA processing, or RNA splicing. Particles composed of proteins and a special type of RNA called small nuclear RNA, or snRNA, play a role in RNA splicing. One kind of small nuclear ribonucleoprotein, snRNP, contains snRNA that can bind to the 5' end of an intron by forming base pairs with complementary sequences on the intron. A different snRNP binds to the 3' end of the intron. Additional introns interact, causing the intron to form a loop, thereby bringing the two ends of the intron together. The large complex of snRNPs, called a spliceosome, then excises the intron and the exons are joined together. The snRNPs are then released.
So these ribozymes, these are kind of a sort of relatively new discovery, because we always thought that all biological catalysts were proteins. But now with the discovery of ribozymes, we found that these are catalytic RNA molecules. Remember what a catalyst is. It's something that assists these reactions. And we talked about how enzymes do that, but these RNA molecules function as enzymes and can actually splice the RNA. So the discovery of ribozymes rendered obsolete the belief that all biological catalysts were proteins. Okay, on to concept 17.4. We've talked about transcription, which remember is converting DNA to RNA. Now we're going to translation, which is converting RNA to protein. And the basic concept is this. So we have what is involved. We're going to see the same three stages: initiation, elongation, and termination. Only in initiation, everything is going to load up on the ribosome. And so here in brown is a ribosome. The ribosome actually consists of two subunits. This large subunit you see here, and then a smaller one down here. And in between these two, sandwiched in between, is where the messenger RNA is going to bind. So there is a messenger RNA binding site, and then we have what I call the parking or parking spots. We have 1, 2, 3 parking spots, and this is where transfer RNA comes in. Transfer RNA does just what the name implies. It transfers something, but specifically what it's transferring are the amino acids. And what amino acids they're going to bring up depends on the codons. So if I were to look up GGC on that genetic dictionary that I showed you, we would find that it codes for glycine. But in order to find the right parking spot, the transfer RNA has to also have this anticodon region. Think of it as a parking permit that complementary base pairs to the codons. So G would pair with C, G with C, and C with G. That's the anticodon. The anticodon does not code for an amino acid. Only the codons code for the amino acid you see here in purple. But it tells the transfer RNA where to park. So notice this middle parking spot, UUU, brings up AAA, which happens to have phenylalanine and so forth. So the reason we need 3 parking spots is because the incoming amino acids are going to come in here. They're going to offload their amino acid to the growing polypeptide chain, and then they move over and exit. So when we look at the details of this, there's really, your author gives us 3 ways to look at the structure of transfer RNA. You can look at this 2-dimensional structure, a 3-dimensional structure, or what I like right here, this nice little symbol used in the text. And notice there's 2 important sites here. There's the amino acid attachment site, which I always think of this kind of like a fishing hook. This is going to hook the amino acid, and then the parking decal, AAG, that tells it where to park. So that's the anticodon, which will then bind with the codon. Remember, the codon is on messenger RNA. The anticodon is on tRNA, or transfer RNA. Here is a computer model showing this whole thing in action. So we have our large subunit and our small subunit of the ribosome. And then we have our parking spots here listed as EPA. That's not the Environmental Protection Agency. In fact, we actually read it backwards because the incoming amino acids are going to first stop here parking at A. Once they offload their amino acid on the P site, which is where that polypeptide chain is being formed, and then they go to E, which is exit. And as they go through this, it's almost like a ribbon through a typewriter type of thing. As they move along, this forms that polypeptide chain, which is the protein. This is a schematic model showing these binding sites. Again, we have the A specifically means amino acyl transfer RNA binding site, but I always think of it as incoming amino acids. A for incoming amino acids. P stands for peptidyl transfer RNA binding site, but I think of it as polypeptide. That's where the polypeptide chain is forming. And then E is the exit site for the transfer RNA. So just like trucks dropping off their supplies, they're going to come in, park, drop off their supplies, and then go out and get more. Come back in. So they go from A to P to E, back out, come back in, A, P, E, and so forth. On and on. They're reusable. There's also on the small subunit a messenger RNA binding site. So again, that's got the codons, whereas the transfer RNA has the anticodons. So let's take a look at this again. And now we see, remember the EPA. So we have the A site here, the P site here. And to start with, in initiation, we have to have everything, again, we have to have at least a transfer RNA loaded on the P site. And then the incoming comes to the A site. And the next amino acid then to be added is on the A site. We add that then to the one that's on the P site. They form a peptide bond, and now you have your polypeptide chain forming. And then those on the P site move to the E site and release the transfer RNAs, and out they go. And this just continues. So again, they just keep moving from A to P to E. Okay. They keep moving from, as I said, A to P to E and off. So once again, there were 3 stages, the same 3 stages in translation that we saw in transcription, but there are different things going on in each stage. Even though they have the same name, there are different things going on. So with the same 3 stages, initiation, elongation, and termination, but what's going on here in translation is different than what we saw in transcription. So here's a clip to summarize that.
The genetic information stored in DNA is transcribed into messenger RNA and then translated into protein. Translation begins when messenger RNA binds to the ribosome. The initial transfer RNA occupies the P site on the ribosome. Subsequent transfer RNAs with bound amino acids first enter the ribosome at the A site. The complementary matching of 3 nucleotides on the transfer RNA, called the anticodon, and 3 nucleotides on the messenger RNA, called the codon, ensures the correct sequence of amino acids. The messenger RNA passes along the ribosome in short spurts of 3 nucleotides at a time. As this occurs, the initial transfer RNA is moved to the E site and its amino acid is transferred to the second amino acid at the P site. At the same time, a new codon is presented at the A site. The initiating transfer RNA, which now no longer carries an amino acid, leaves the E site and the next transfer RNA with a complementary anticodon enters the A site. Each time a new codon sequence moves into the A site, a new transfer RNA brings in an amino acid. The old transfer RNA paired with the previous codon is passed to the P site and then to the E site as the amino acid it carried is transferred to the growing amino acid chain. As the ribosome proceeds down the messenger RNA, a stop codon is finally encountered. At this point, the ribosomal complex falls apart and the protein is released into the cell.
Okay, so once again, looking at the details of initiation, this is now on page 108 of the course outline. And the initiation stage of translation brings together messenger RNA, transfer RNA with the first amino acid, and 2 ribosomal subunits. So we're all loading up together. So we see if we look up and a close-up here of the codon-anticodon pairing, we see we start with that start codon, right? AUG, we start classes in August. There's your start codon. This is on messenger RNA, which is bound to the small ribosomal subunit. And then the anticodon is going to be complementary. A with U, U with A, G with C. Remember, no T's in any of these because whether it's a codon or an anticodon, it is RNA. So there are no T's in RNA, only on DNA. And so there's your parking decal, and it knows where to park, so it's going to be parking on that P site. It's going to be bringing up the amino acid methionine, which is what you would find if you looked up the codons AUG or the triplets AUG in that dictionary. You would find it codes for methionine. So again, one start codon codes for methionine. So does that mean that every protein begins with methionine? It does initially, but there are modifications later that change that out. So it's just the initial setup. But notice the E and A sites are still open. So this is the translation initiation complex step 1. Now, the next incoming, now we're getting to step 2 where we're looking at elongation of the polypeptide chain. And during elongation, amino acids are added one by one to the preceding amino acid at the growing chain. So here we have the incoming, again, going on the A site. The incoming amino acids load up on the A site, and then they're going to offload this amino acid onto that growing polypeptide chain. And then they are going to move from the A to the P. Everything moves over, and one falls off. So the E site moves out. That opens up the A site. The new one comes in. The one on the P moves to the E, and so forth. It just continues as you saw in that animation. And the polypeptide chain moves along until it reaches that termination point. So in this case, what is the termination point of translation? Termination occurs when a stop codon in messenger RNA is reached. So remember, we had one start codon, AUG, and we have 3 stop codons. These codons do not code for an amino acid, right? All they do is they donate a protein that is a release factor, and this release factor then causes the addition of a water molecule instead of an amino acid, and that releases the polypeptide chain, and translation assembly then comes apart. So let's take a look at that. So once again, in the termination, we have a stop codon right here. It's either going to be UAG, UAA, or UGA. And the release factor then will be brought up and attached, and it just blocks that site. And since it's blocked, everything's going to be released. And now we have our free polypeptide chain. These transfer RNAs can be reused. The subunits for the ribosome can be reused and all of that. And here we have termination. Again, everything can be reused. And now you have the polypeptide chain. Now, when you need a protein, you usually don't need just one little polypeptide chain. Let's say you've been in an accident and you need blood. You need proteins to make blood, right? You need hemoglobin, for instance, to make red blood cells. So this has to happen very, very rapidly. And so it has to happen with hundreds or thousands, really, of amino acids simultaneously. So how do you do that? And the way that we do that is through something called a polyribosome, or polysome for short. And polyribosomes enable a cell to make them many copies very, very quickly. So as you see here, we've got your DNA shown in blue, and then you've got many of these polymerases moving along. Remember, these are RNA polymerases. Why? Because they're making RNA, not DNA. They're all reading the DNA at the same time simultaneously, and each one then is producing in red here. This is a blow up to show you that that's the messenger RNA, which then is bringing up the complementary or the proper amino acids. So you see each one of these ribosomes are pumping out a separate polypeptide chain. So many of these are happening, and this is an actual micrograph up here of a polyribosome. So many ribosomes loaded on the messenger RNA and simultaneously pumping out the correct protein. So, and you have different proteins being produced at different places as well. So all of this going on very rapidly. So actually, it's something like hundreds of amino acids are added per second, which is amazing. All of this is really, truly amazing that it's going on right now in your cells right now. As you listen to me explaining this, you are pumping out proteins using this mechanism of protein synthesis. So at this point, a lot of people are kind of going, well, this sounds pretty complicated. How am I going to learn all of this? The best way to learn it, I think, is to sort of sketch it out and make sure you include all the players. So this is another one of these parts of this course that lends itself nicely to a sketch. So I would practice learning this by sketching out, showing a nucleus, what's going on in the nucleus, specifically transcription. So show your gene region of DNA, show how it's open here, make sure you identify that this is RNA polymerase, and you can show the pre-RNA transcript forming here. But remember, you have to include the RNA processing. You have to include the fact that your introns are being cut out, your exons are being spliced together. You're adding the poly A cap, or the poly A tail, and the 5' cap. 5' meaning the 5 carbon end. And out it goes into the cytoplasm. Out in the cytoplasm, this is where translation occurs. And so here is your ribosome in brown. You've got your 3 parking spots here, your EPA, and remember your transfer RNAs are going to bind there, and they include, make sure you recognize they include the anticodon as well as the amino acid, and make sure that you show the codons on messenger RNA. And then practice with some of these base pairs. And here's a good practice for that. This is over on page 109 in your course outline. And given the following base pair sequences, and I'm going to give you a minute to practice this. So in this one, here's your sequence TAC GCA. Convert that to a DNA replicate. And remember, in DNA, yes, you can have T's, but then you want to convert this gene to messenger RNA codons. Remember, there's not going to be any T's here that will be used, and then convert messenger RNA codons to transfer RNA anticodons. Again, here you should only have U's, no T's. So take a minute and do that, and I'll be with you in just a minute. Okay, let's try the first one. So T, converting, remember when you see a gene template, a gene is a region of DNA. So you know this is DNA. Also, it has T's. So when we're making more DNA from this DNA, T brings up what? A brings up T, C with G, G with C, C with G, and A with T. So there's the DNA replicate. Now going in this direction, taking that same gene region and converting it to messenger RNA codons, T brings up, A is okay, but A brings up what? U, right? C with G, G with C, C with G, and A with U. And then now we're going to convert the codons to anticodons. A with U, U with A, G with C, C with G, and so forth. And so here is where we get confused sometimes. If I give you a gene region, a piece of DNA, as you see here, and ask you for the codons, most everyone's okay with this. But what if I give you this same gene region and ask you for the anticodons? Now you have to remember the middleman. You have to first convert the gene to codons, the codons to anticodons, and that's how you get the anticodons. Or if you want to skip the middleman, you can just take the gene, change the T to U's, and you'll have the same thing. But the point is that every one of these is a little bit different. So you have to be careful with that. Okay, at this point, to summarize everything, hopefully this is working, and we shall watch this BioFlex. Not sure if this clip. Nope, sorry. I changed it and forgot to take that out, so I will have to take that out. I have a different one coming up. There it is. Okay, so still looking at your course outline, page 109. At the end of all of this, once that polypeptide chain is formed, there are post-translational things that have to happen to it. And that's all in chapter 18. And we are, even though chapter 18 is in your course outline, we are skipping that. So don't worry about chapter 18 because you'll get to that in other courses if you continue on in biology. But I did want to just mention briefly a little bit about these post-translational modifications. So that polypeptide chain, once it's finished, it will spontaneously coil and fold into a three-dimensional shape. Because remember, proteins are all about shape, right? We've talked about the primary, secondary, tertiary, and all of that. And sometimes they need some other modifications. And again, that's what chapter 18 is about. Some polypeptides are activated by enzymes that cleave them. Remember, that means that they might need to be split, or others come together to form subunits. So here's a little bit about that. Oh, that one's not working either. Huh. Okay, sorry. Thought I fixed that one. Not sure why these are not working right now, but moving on. Okay, so the last section on page 109 is dealing with targeting polypeptides to specific locations. And one thing I didn't mention about ribosomes is that there are two different populations of ribosomes. There's free ribosomes, those are in the cytoplasm, as you see here, or cytosol as it's called. And there are bound ribosomes. Those are the ones on the rough ER. So remember we talked about rough and smooth ER. The rough gets its name because it had ribosomes all studded on the surface, as you see here. They both have the same job. They both make proteins, but free ribosomes mostly synthesize proteins that function in the cytosol. So, for example, those might be proteins that form form digestive enzymes that are needed in, say, lysosomes in order to do cellular digestion, or they are bound ribosomes, or the bound ribosomes, that is, are secreted, make proteins that are secreted from the cells. So those might be maybe proteins that are needed to make hemoglobin outside to make the red blood cells, something like that. But either way, ribosomes are identical and can switch from free to bound. And that is our wrap on protein synthesis. Now, since my video, that last summary video clip did not play, you can certainly go on YouTube and search, and there are so many videos on protein synthesis. So take a look if you still need some more help with that, and also see me on chat or email me if you have any questions. So I know this is a lot, so practice sketching it. That's the best way to learn it. And let me know if you have questions. And I will see you next time for chapter 20. Have a great day.